And now, an exclusive sneak preview of a recent X-Talks webinar, Tumor Immunogenicity Biomarkers, the potential of Immunoscore IC in drug development.
Let's get into the principle of Immunoscore IC. This test was developed taking into account that the current capability of biomarkers used to predict the response to immunotherapy was pretty low. Today, the main marker is PDL1, detected by immunohistochemistry and analyzed through various methods like the tumor positive score or TPS and CPS, but basically the idea is to quantify visually the expression of PDL1 by tumor cells or immune cells. We know that PDL1 is definitely a good biomarker when we are talking about prescribing anti-PD1 or anti-PDL1, but with very strong limitations. We believe that, in fact, the most effective way to predict the response to drugs targeting the PDL1, PD1 axis would be to look at PDL1 for sure, but also to make sure that the tumor was infiltrated by lymphocytes, especially T lymphocytes, and probably CD8 positive T lymphocytes. That was presented by various authors, and we added to this assumption that it could be interesting also to add a level of complexity by analyzing the proximity between the PDL1 positive cells, whether they are from tumor or immune origin, and CD8 positive cells. We had to build some innovative tools to do that, and this is exactly the purpose of the Immunoscore IC tests, who leverages digital pathology in addition to immunochemistry to determine a set of variables that can be combined into this Immunoscore IC score. So, in summary, we will be able to look with the test to look at the densities of PDL1 positive cells, CD8 positive cells, but also the proximity between them or the proximity of PDL1 cells to other PDL1 in the assessment of the clustering of those cells. Same thing for the CD8s, and combine all this thanks to advanced image analysis. There are 4 steps to the test. The first one is a dual staining immunohistochemistry assay, where proprietary antibodies are used to stain CD8 and PDL1 biomarkers. Then the stained image, the stained sections are digitized with a slide scanner. Then the image analysis can be done, looking at the different parameters that we will get into a bit more granularity, and combine them into a classifier to determine the score of the tumor. So first step is the dual staining and cell detection. We can see on the left an image where the CD8 positive cells are stained with detected with the ultraviolet detection reagent. Same thing for the PDL1 positive cells with DAB. So it's quite direct double staining with 2 primary antibodies from different species that can be detected simultaneously on a single section. The test needs only 1 section, and this dual staining can be the sections can be digitized, then analyzed with a software to quite easily detect all the different cells, but also get a very precise detection of the CD8 positive, PDL1 positive cells, and we will see some analytical validation later on. So concerning the image analysis, there are different things that can be done. The first one will be to detect where the tissue is present on the image, and within this region of interest, identify the tumor area where eventually we will be looking for the cells of interest. The cell detection, we have quickly touched it. The critical aspects of the cell detection will be the accuracy of this detection, so that we do not get any false results. And on the bottom right, the addition that we bring with this assay is to look at the proximity between cells. This is fully automated, and the principle is to take any individually stained cell, define its centroid, and look for the presence of another cell of interest within a radius of 20 microns. And concerning the classification of the tumor, the idea is that we can get plenty of parameters or variables out of the image analysis, among which the cell density of CD8 positive cells, PDL1 positive cells, the clustering of each of those populations, and also, of course, the proximity between them. All those variables, after univariate analysis, were included into a Lasso-Cox model that proved to be very helpful for the prediction of the response to immunotherapy, and Jérôme will cover it in a moment.
To view the entirety of this webinar, visit the link in the description and register on xtalks.com, completely free of charge.